University. So in the presentation order, I'm going to have this presented. You can just go to the next slide. Yeah, we said Professor Kibet is based in Egerton University, Department of Chemistry, and he's a specialist in computational chemistry. To introduce, we're looking at marijuana. Everyone here has been talking about the safety by looking at the soil analysis, focusing on heavy metals. And then our second presenter was talking about water, how water can be purified or made safe for us to take. Our work here, we're looking at air, focusing on air, how we can make this air that we breathe safe for consumption or for breathing. Now, marijuana is one of the most, or the second most abused drug substance after tobacco, as reported by the NIDA 2020. And most of us here, we must have come across or interacted with people who use marijuana or probably smoking marijuana. Now, of late, we have an exponential increase in the trends by which this drug substance marijuana is being used, because some nations or some countries, including Canada, have legalized its use. But in Africa here, or in Kenya, probably, to be precise, we don't have legalization for it to be used. So it's limited. But we find most people, including the students that we teach, sometimes they smoke this marijuana around. So when it's being smoked, or the smoker is using it, he becomes the mainstream smoker because he takes the mainstream smoke. Us who don't smoke, we become passive smokers. Now the danger comes in that the smoker, to some extent, is safe, because if he's using a cigar that is well designed, it has filters. Now these filters are going to filter out some of these toxic compounds that are liberated or generated during smoking. But you, as a passive smoker, when you pass around and inhale that smoke, now you directly take those toxic compounds which have been generated by this smoking or burning. They get into your body, and probably they're going to cause harmful or toxic reactions in your body. So this marijuana is of interest because it is a plant which is complex, in that it has been identified to be having more than or close to 500 compounds and 60 cannabinoids. Now these cannabinoids, when they get burnt or when they are combusted, they generate up to over 2,000 chemical compounds, some of these which are possible toxicants. Next slide. So the objective of this work, we looking at or describing the mechanistic degradation pathway of CBD and THC. Those are cannabinoids in marijuana. And then we shall look or compute the global energies and entropies of CBD and its derivatives using Gaussian computational platform. And then third, we shall look at, or we shall perform we have already done that. We shall perform, we perform geometry optimization of the CBD using Gaussian computational program code. Then we calculate the toxicity indices of CBD and THC and their derivatives using the Hyperchem computational program. Next slide, please. Now, why we justifying this study? As we said, marijuana smoke leads to acute vascular and endothelium dysfunction among the users. And then it has injurious psychological and physical effects, that is psychiatric disorders, and disruptive effects on pulmonary and respiratory systems, which is caused by this uncontrolled use of cannabis. Then despite these non-toxic effects, we have seen marijuana getting legalized in some nations for use both in medicinal and some in recreational purposes. So we need to look into this compound marijuana. Then lastly, the consumption is also increasing. I already mentioned that, and this one gives us critical or raises critical concerns on the toxicological process that are induced by this product, marijuana. Next slide, please. So the methodology we employed here, the theoretical and electronic DFT calculations were performed using the Gaussian 09 computational code as described by those two authors. Under this, we had the B3 LYP, that's Becker 3 parameter Lee-Yang-Pearl hybrid functional level, hyphened with the 6311G basis set when we were conducting the calculations according to the order there. So the initial structures of these cannabinoids, THC and CBD, were optimized to their global minima using the Gaussian threshold convergence criteria at a root mean square force of less than 0.003 Hartree per angstrom. All these calculations were single point energy calculations, which are done within the temperature range of 623 Kelvin to 1023 Kelvin and a pressure of 1 atmosphere under conditions that simulate the actual smoking of marijuana. Now, next slide. The enthalpy calculations, we did the calculation using this formulation where we have enthalpy changes given by the summation of enthalpy of the products less the summation of the enthalpy of the reactants. Under that, we have defined the factors in that equation, where we have the delta RH being the change in enthalpy of the reaction, H called the correction to the enthalpy, and epsilon not the sum of the electronic thermal properties. Then we went ahead and used hyperchem computational code under the quantity structural activity relationship, which are models which are used to estimate the toxicological profiles of these two cannabinoids and their subsequent radicals. Next slide, please. So the first, we described the thermodegradation pathway. Under this is a proposed mechanism by which CBD is going to undergo thermodegradation to give it to form the respective products. So first, it breaks down to give us the hydroquinone CBD, which is a radical. Now, this radical is very unstable. It reacts with hydrogen. This hydrogen probably in our body system, in the body system, to form 5-pentylbenzene 1,3-diol. This is common known as limonene. Then the other radical there, 3-methyl-1,6-prop-1-N-2-ol cyclohexane 2,1-helium, that's a carbocation, which is also a radical, very active in the body system. This also gets converted to 1-methyl-4-prop-1-ene cyclohexane 1-ene. That is commonly known as olivetol. Those two formed products, once they get into the body, some, for example, limonene, this one has been reported to be a product that leads to generation, genosis of cancer. Then olivetol, the same, it's an irritant. It has been reported in the body, so it's not those are not safe products or compounds once they get into the body. 10 minutes are up. Okay. So next slide, please. Let me just be very brief. So the thermodegradation of THC resulted to the formation of all these compounds. These compounds, as you can see, we have quinones here. We have benzoquinone, CBD quinone, which are the products not safe for human body. They can also get converted to these polycyclic or aromatic hydrocarbons, naphthalene, anthracene, benzoapyrene. All these we know that they have been reported to be cancerous. So once you consume that smoke while smoking, or if you are a passive smoker, probably they're going to cause so many effects in your body. Next slide, please. So the next slide here, we're describing the computational enthalpy changes. Next slide. This one, I think it's a table showing the energy changes that we obtain. Please, next slide. It gave us a curve, and this curve shows the enthalpy rise, how the enthalpy changes with the increase in temperature. But towards the end, we can see that there's a slight fall in the temperature. Probably that slight fall, we were to explain to it, probably because of the number of carbon bonds, or the number of bonds within those compounds having reduced, and then probably leading to that small drop in temperature. Next slide. So we generate the geometries of cannabis derivatives. If you look at the first A there, the figure A shows the input structure that we put into the computational software, and it was able to optimize this, to give us the most stable compound of CBD. For CBD to get optimized, it took about 40 optimization steps at an energy of around 967.9 there. While THC, for it to attain stability, it took about 68 optimization steps, as indicated by those optimization step diagrams, are those energies there, showing that the stability of THC in the body system, it will require so many changes or reactions for it to become very stable, for it to be in your body system. Next slide, please. So on the other diagram here, we have the molecular orbitals for these compounds and their energies. So the first one there shows the the molecular orbital for this is the highest occupied molecular orbital for CBD at an energy of 0.340 electron volts. On the other side, we have the highest occupied molecular orbital for THC at that is 0.218. On the other side, we have the lowest occupied molecular orbitals, which was orbital number, that is 40, no. The figures are not so clear, but those were the structures we obtained at the different images. And this highest HOMO and LUMO band structures gave us a band gap energy of positive 0.157 electron volt, while in THC it was 0.214 electron volt. Now, what does this one tell us? We're talking about the electronic structure of these compounds. We have the band gap energies, like how the electrons become excited in the human body once they become smoking, and they're going to jump from one band to another. Now, when we have a larger HOMO-LUMO band gap, that means this is fairly stable compared to a lower band gap energy, since a lower band gap energy means the molecule gets easily excited the electron in the molecule easily gets excited to jump to the next band, making that compound or molecule so much unstable. So CBD here becomes so much unstable compared to THC. Next slide, please. So this led us to the generation of the electron density diagrams for both CBD and THC, as shown in that, where we have the blue regions showing the sites of positive values of different densities, and the blue regions showing us the positive sites, while the red regions will give us the negative sites, and how this electron density or the electrons are moving along that molecule. Next slide, please. This resulted to the contour maps, which are as shown in the next slide here, please. The next slide is supposed to show us the contour maps for these two compounds. 80, please. Next slide. Yeah, those are the contour maps for both CBD and THC.
Kindly
next
conclude.
Slide.
Conclude.
On the toxicity estimation, we used the QSAR, that's Hyperchem, to estimate the log P values. And these ones, most all these values were of more positive values. But we know that log P, when it is negative value, it shows that this compound is less reactive with or it's more reactive with the aqueous phase and not the lipid or solid phase. But when we have more positive values, that reflects that this compound are more reactive with lipid. Now, all those values were most positive. Actually, all of them were positive, indicating that these compounds are likely to react with the lipid systems in the body. And once they do that, they're going to affect the DNA structure. This one is the basic step where we start the process of carcinogenesis. I think finally, if we conclude, the next slide is giving us the conclusion of that. We can say that this thermodegradation of CBD can lead to the formation and release of toxic compounds, and the QSR can enable us to estimate the toxicity of these compounds. Therefore, CBD can thermally degrade into potentially toxic effluents, which render it equally toxic even if it finds its application in medicinal use. Thank you.
Thank you so much. Give him a clap for the next presentation. I think he has really done well, although he has really eaten our time. Actually, 10 more minutes. So I'm going to open the session for any other questions. 2 burning ones, but it seems like I did the basics of bioinformatics or biology. And there was something we used to call as Lipinski rule, where you have the admit component, and T comes last as toxicity. Did you look into that? Because I want to believe, by the time you take something, it gets absorbed, it has to be detoxified, it has to be metabolized and all that. Did you look into that?
Come up again. I think there's something I'm not getting clearly.
Admit. Did
Admit,
you do
yes.
that analysis? Yes.
So, your question is?
Did you carry out admit?
Now, this one is computational analysis.
Yeah. Still the same.
Yeah.
Still the same. I'm asking is purely computation.
Now that one I cannot tell you that I've used it.
All right. Thank you.
I'm not familiar with that.
Thank you.
Yes.
Any burning questions, please? From chemistry, from biology, from physics, from mathematics? I think well done, Prof. Do you have any maybe burning question?
So my question is coming out of curiosity.
Yes, Prof.
Allow me to just formulate it with the statistics that are just coming from my head. But for every probably 1,000 people that abuse that drug, 3 to 5, it helps them. There's a small percentage that it actually helps, but it's very, very small. So it has medicinal ability to, because the idea is you're looking at the electron, and the brain is an electrical system. And so there are some people who may have gaps, and so it helps them. But the negative effect is what you are pointed or alluded to because of that gap. So have you seen from the literature what they are saying about this small percentage that it actually helps? Because that's the reason why most of the regulation has actually come in, because there are people, very, very small percent, that it helps.
Thank you, Prof. Now, from literature, this small percentage, it helps those people with cancer. It's mostly used as a painkiller when to manage the cancer patients when they are in pain. So probably the CBD, that's why it finds major use as in a medicinal application. But now not all of us have got cancer. So if I was passing around, for when you are in pain, you can do anything so that you can feel, you can get relieved. So for cancer patients, already they have the cancer in the body, you need to manage the pain for them to feel comfortable. So probably that's where it finds its application. But nowhere else we have found it applicable in medicinal purposes, apart from in cancer patients. So if I come here, I'm smoking, I'm endangering your life, because you as a passive smoker, you're going to inhale the smoke directly. But as the mainstream smoker, I'm having filters that are going to control the use of the compounds that I'm taking in.
Thank you so much. So that maybe implies that it works like an antioxidant, because I wanted to also ask a question.
Okay, thank you. Now this is the first presentation which is close to mathematics. I've sat here since morning and I've just been hearing some chemistry things. So just a quick one. I saw some p-values there.
P-values, yes.
Yes, the toxicity, I
Yes.
think estimations.
Yes.
So what were they?
Now those are log P values, the octanol water partition. Now when we estimate them using the QSR, they are supposed to tell us how the body will interact with these compounds. Now if you have a negative value, then that tells you that this compound is hydrophilic. It cannot, it much interacts with the aqueous part of the body and not the lipid part. But when it's highly positive, this is more interactive with the lipids. That is when it's more positive and less, more positive actually. So most of these values you've seen there, they are positive values. Simply they're telling us that these compounds highly interact with the body lipids. But remember, these body lipids are the major components that are forming the body DNA structures. So once it has started interacting with this, it's going to convert or change its function or its growth. So mutagenesis now strikes in.
Okay, well explained. And what about the confidence interval? You don't have a confidence interval maybe from negative something to positive something?
Now, the acceptable values should be not more than 2.
Okay.
But most of the values, they are close to 2, and others were beyond or above 2. Okay, yeah. Thank you so much for listening.
Let us have the PowerPoint shared for Evan Sonyancha as he asks this one question.
Maybe mine was on the positive aspect. Can we by any chance have a component of marijuana in sedatives?
The
Sedatives.
sedatives.
Yeah.
Marijuana and sedatives.
No, I'm saying, can it by any chance have a component of marijuana or cannabis sativa in sedatives?
These are painkillers. What's a sedative?
Maybe drugs that are used to take somebody to sleep.
Not all. I cannot say about that because we have not analyzed any drug to check whether it has this cannabis present in it. But there are those drug substances which are or where we have cannabis declared that it's present, right? So unless we do analysis and know which drug you're talking about exactly. Thank you.
Thank you, please.
Okay.
Computer-based, purely conversation.
Habari ya jioni wenzangu. Habari tena. I know it's after lunch and you're a bit heavy. But let me take this time to once again go to these herbs. We've been looking at herbs since morning. But my area or my angle of attack of these herbs, it's like we can go beyond the lab. Because most of the work is being done and left in the lab. Beyond the lab, we leave it for, how do we call them? Big pharmas. The pharmaceutical companies that are quite huge and are well endowed. To begin with, my name is Onyancha. I'm an organic chemist specializing in natural products, drug development, especially from the herbs. And finally, I have an expertise in AI in drug development. I think that's the latest workshop that we attended this month. This is part of my PhD work. which I'm being supervised by Dr. Nyabaro, Dr. Kenanda, and Dr. Nyancha. I've picked a bit of my research and zeroed down to these two compounds from Indigofera erecta. Otherwise, I'm working on two plants, Cichus peciorata and Indigofera erecta. So far, I want to narrow my discussion today into butyronic acid and the frosine from Indigofera erecta. Welcome. Let's go to the next. Good. Now, Indigofera erecta, it's a common herb that is kind of a nuisance. It grows where the ground is disturbed. And for those who do farming and maize and all that, this is a common nuisance. It's a tall, relatively tall, not quite tall. It's used in South Asia to produce the color we call indigo. That is a color for jeans. That's blue, deep blue color. But locally, like here in Kisii, we call that plant omocheo. For those who understand that dialect, omocheo. Locally, these are some of the uses: epilepsy, nervous disorder, diarrhea, healing wounds, as an apophysiant. Equally, it fixes nitrogen. The next. That is the plant I'm talking of. Tukiwa wadogo tulikuwa tunachapwa na hiyo viboko ya hiyo mti. Very, very, very slender and tall. I got it ground, dried, then extracted, and then some sections of the extraction I loaded in a column, ran the column. The far end, some of the portions I was getting after cracking that column, running it several times, and using TLC to see what I'm getting out of this. And then finally, these are some of the pure compounds I was getting down here, the small bottles. Some were white, some were crystalline, some were pinkish, and all that. How do we know that it's pure compound? We use TLC. When you get one spot, you are done. After getting these pure compounds, we run the NMR. Let's go to the next. We run the NMR, the mass spec, and all that spectroscopic techniques. And those techniques assisted me to come up with this structure as one of the several compounds I got from this plant. The others in Meka Kando, I just wanted to deal with only these two. And in this molecule, the most active parts are in carbon 3 and carbon 28 there and carbon 22 there. And that is where most of the chemistry is right now. But there a big pharmaceutical company has this molecule you're going to see. Let's go next. After getting the spectrals and getting the molecular structure, I realized this compound exists. But it exists in other plants, like in China. They get it from the beech bark, the bark of the beech plant. But this is the first time it's been isolated from Indigofera erecta. And this is a very prized compound after searching some literature and data banks. I discovered this molecule is almost in the middle stage of being processed as a cancer drug. But it's yet to make it to the final end. But it's relatively active. It's a pentacyclic triterpenoid. It's rarely soluble in water. And the good thing about this molecule is it has very limited side effects, unlike these other common cancer drugs. It targets the cancerous cells mostly. That is a unique part of it. Although the shortcoming is it's not soluble. It's not really soluble in water. At the same time, it has very good anti-HIV activity. So it's a stone that kills two birds. At the same time, can be used in malaria. That is from the literature after getting this molecule. My main aim was to test antibacterial activity, which I did. But these are quite interesting after discovering the molecule. Next. The second molecule is the frosine. It's a flavonoid, derivative of a flavonoid. Equally, I got it the same way. It's one of the molecules, one of the fractions. I got this. It's equally an interesting molecule. After discovering that I have this molecule, I found it in literature. It's equally being used for cancer, but not as a betonic acid. Next. These are the NMR, the carbon and proton NMR that assisted me to come up with that structure. And now that is our chemistry. Next. Next. So equally, these are some of its bioactivities that are online. You can easily get this from, by the way, we have the drug bank, we have the molecule bank, banks of molecules, bioactive molecules, and all that. So from this, Indigofera recta has two anti-cancer and bacterial and antivirus compounds that are advanced in the drug making. Next. After isolating this, as much as my concern was about bioactivity against bacteria, and after getting all this information and these compounds are now a hot soup, a hot cake. What do we do next? I did my master's in natural products some time back. I got my paper. Everything was forgotten. It was thrown on the shelves. And now I'm back with my PhD. I've gotten some interesting compounds. What next? And there are several herbs that have such a potential. At the same time, we have AI that assists us to get bioactivity by using by using dry lab. In my case, I was concentrating on wet lab. Dry lab is where we carry out all these tests online using the machine learning, that is the AI. You get to train this AI, you identify a module, train it using a given data bank of the type of molecules you are dealing with and the results, whether they are active or not. After training it and doing all the querying and whatever, and that is now in mathematics. Statistics is quite in use there. But who are programming, you come in. And then, after training this machine, now you can run any molecule. You can get the ADMET online, that is solubility, blah, blah, of drugs. If it's antiviral, you can get it. Then you can generate homologous compounds. If there is one compound that is bioactive, you can generate related compounds, whether it's structure or activity. If equally these compounds, you can run them online, such that you can have potential molecules before going to the wet lab, where you're going to do now the real culturing and testing. All this I'm saying that you can do, it's to the advantage of big pharmaceutical companies. Because developing a molecule up to a drug, it takes hundreds of millions or even billions for that drug to be on the table. Do we need to be waiting that? No. There are alternatives. There are alternatives. Next. There are two feasible routes. As I said, the first one is quite capital intensive and time consuming because it involves a lot of testings, clinical trials, a lot of money, volunteers, blah, blah, blah. The second one is go the supplement or nutraceutical way, where you can standardize your product and then you solve the ailment. Because some of these terminal ailments don't have drugs. Well, there is no treatment. You just manage, like cancer, HIV, and all that. So equally, with nutraceuticals or supplements, we can equally manage. Just like we are managing with these other plants, these other drugs. But this requires money. It requires money, and that is where our funders come in. And that's where we can appeal to the research department to consider this. We identify these herbs, domesticate them, produce them mass, we do the dosage, and then to avoid these long time-consuming processes, we go to the nutraceutical way, which equally saves life, irrespective of the many questions from the big pharmaceuticals. Equally, nutraceuticals save life. Next. And that is the proposed way of getting this knowledge off the shelves and back to business and back to wellness. I'm using the term terminal illness in quotes because who knows, after getting these nutraceuticals, it may not be terminal anyway. Thank you and be blessed.
Yeah, thank you so much, Mr. Nyancha, for the nice presentation. Yeah, we will open the floor up for questions in terms of the work that he's doing. So, any question?
Thank you, Mr. Nyancha, for the wonderful presentation. If I got you right, you indicated that you are working on antibacterial activities of the extracted compounds that you've detailed more about. Now, I'm wondering, you said at some point some other compounds that you extracted were forgotten and you proceeded with it too. Now, I don't know if it is out of some preliminary and microbial activities that you assayed and you found the two to be more active than the ones that you forgot. And if that is the case, did you consider to check if the forgotten compounds had some synergistic activities, maybe more than the two that you've measured on? And number two, during your material correction, I don't know which method that you used to correct your materials. And lastly, plants respond differently to different seasons. I don't know if you've checked throughout the growing here season, which season could be appropriate for correction of material so that you can obtain the highest concentration of the compounds that you are measuring in. Thank you.
Thank you for that concern. Maybe you got it wrong. The first instance about, that's the first question. I'm responding to the first question. I never left the molecules. I picked only this, but they have around 17 of them. It was my choice to work on this because they've never been isolated from this plant. The rest are there, like in Irubin, I saw indigo and all that. But these ones were unique. Were unique in that they've never been isolated from these plants there before. And those indigo and all that, they are equally anti-cancer. So this plant is like, it's a pot of anti-cancer compounds. Okay? They are part of my thesis, but I'm not presenting the thesis. That's why I said it's a bit of my thesis. Do I go to the second one?
I mean, I was asking if your studies on antibacterial activities, you're saying the two that you are measuring in are unique in that they have never been isolated. I'm wondering, have you done your preliminary assessments out of the 17 and you found the two to be more antibacterial than the others, leaving aside being anti-cancerous? The other concern was like now you are extracting these compounds from your medicinal herb. Which season of the year?
I got your second question.
Okay.
It's one of the first one. Maybe I can repeat in answering. All these compounds have done the antibiotic tests, the assays on antibacteria. I've done all of them. And that was my main focus. Some are moderately active, they can inhibit to an extent compared to erythromycin, but not quite. These ones surprised me after looking at the literature that they are anti-cancer. I was not dealing with that cancer. And my presentation is, why do we leave what we've done on the shelves? We can go all the way. So on antibacteria, they are all bioassayed, and I have all the information. Only that I decided to work on this, only this presentation. Secondly, yeah, I know the content, phytochemical contents of a given plant depend on the day, the season, even the time, even the way you handle it in harvesting. We took care of that. Consistently, we picked a given season when we did that. And we were not trying to compare on the amount and all that. That's another area, especially when we come to dosage and on how to manage these plants. I think that should be now when we are commercializing. But basically, I picked it at the same time, and these are the same herbs I'm working on. Maybe next time, we'll change the timing. We'll change the season. But I know at the age of around 2-3 years, that's when they bloom, and the content of these phytochemicals is high, quite high.
Okay. Any other question?
Yes, Nyancha, a very good presentation. Just on the first compound, which is butanoic acid or butyric acid. It has a very good biological activity, but it's not soluble in water. How do you proceed with it so that it can be useful on the side of medicine?
That's a very good concern, and by the way, that is why it's not yet hitting the market. In vitro, it's very good. In vitro, but in practical, it's a bit tricky. But I can attribute this to the isolation. When you work on a molecule in isolation, and in nature it's in a combined state, there are some other compounds maybe that help in solubility, especially when they are digested in the stomach. We don't know what happens when a patient takes the whole plant. Several chemical reactions take place, and we have several products in the stomach. The fact that the plant is still being used for tumor and all that, and it's effective, there must be something the isolation does that really reduces its efficacy. Maybe when they combine several of these anti-cancer compounds, I'm not sure about the mechanisms, but somehow they work when they're in combined state. But as a molecule, it's a no-go zone for solubility, very low solubility. That's why they are modifying at those carbons, carbon 3, 20, to add some moieties that can pull it into solution to assist in solubility. Sawa?
Thank you. Thank you, Onyanja, for that thought-provoking remarks you have actually made regarding why we putting this information to gather dust. I think it's high time to look into it, tap into it for that. But then maybe a stupid question, have you patented that? Because if you're going to publish it before you do that, I'm sorry, it might be gone. Then too, maybe I'd request what could be the recovery rate. For example, you have 10 grams of your plant X, and how much of that are you actually getting? And it's also true that, as you alluded, with the fact that they are working, there could be synergism effect because it doesn't work on its own. You need perhaps to do an in silico studies, by having that specific compound of your interest, you can be able to assess if it actually has the activity. If it has, then you can go ahead, maybe do artificial synthesis, then actually be able to check whether it actually has that. It can still be a plant, but if it has a good impact on in terms of the cancer treatment, I guess it can even be done artificially for that purpose. Maybe those are the suggestions I could be able to have now, not a question per se, so that you can actually be able to respond. But then when you are doing all these things, again, it's important that maybe before you conclude that it has cancer activity, you also do a detailed study regarding that. I don't expect maybe you do MTT assay alone and then you tell me that it actually has anti-cancer activity. I could have asked maybe what other bioassays did you really do to really confirm that it has that particular activity. So thank you so much.
Maybe as a conclusion, some of these molecules, by the way, quite a number, are available online. There are data banks like Zinc, PubChem, ChemBill. Like PubChem gives the molecules and its bioactivity. If you go in, it can give you all the bioassays that have been done and certified. Like this molecule, it has around 6 cell lines. And cancer, breast, whatever, skin, all that documented. And you can purchase these compounds.
Yeah,
But we are lucky, we are having these compounds in the plants around us. That is a good bit of it.
And also think of, making use of the liposomes. They be able to solubilize your compound, and perhaps it can actually be able to work. So it's all about bringing brains together and thinking out of the box, and you're able to have a solution.
Sure.
Yeah. Brian, I want to request that in the meantime you share the PowerPoint for Meshach. I think is our last presenter. One more. Yeah. I think, are we done? Are we okay with Evans? Any more questions? There are no more questions. Mwangushie moja. 1, 2, 3. Ijashika vizuri. Ibutu mwangushie. 1, 2, 3. Good. Thank you. Meshach, time's not on our side. I think I've taught you and I know you are summarized. Do your thing in 5 minutes, if you can.
Good evening. Yes, now I'm taking you to biology, the medical side. Yes, my name is Meshach Twea Mwega. In that pamphlet they have said I'm Dr. Meshach, but I'm not yet a doctor, but I'm a PhD student in medical microbiology from this university, and I'm doing my research from the Walter Reed Army Institute of Research. I'm going to talk about computational analysis of a hypothetical protein from a jumbo phage. A jumbo phage is a big phage that has more than 200,000 base pairs. That's a phage that are isolated. So for a bacteria called Pseudomonas aeruginosa. Pseudomonas aeruginosa is a clinically relevant bacteria because it has become resistant to several antibiotics. When a bacteria is more resistant to more than one class of antibiotics, it's called a multi-drug resistant bacteria. There are so many mechanisms, I don't want to go into them, that makes this bacteria to become resistant to a lot of these antibiotics. And the World Health Organization has placed Pseudomonas aeruginosa as one of the priority categories of bacteria that should have alternative interventions, because the antibiotics that are being used in the hospitals now are not working at all. If you go to the next slide, the implications of this Pseudomonas aeruginosa in a human being are throughout the body. It can cause infections in the meningitis. It can cause infection in the lungs. Can cause infections in the urethra. It can cause infection at every part of the body. But the problem is that antibiotics that we are using now, they are not working at all for them. Next. So, this is the problem. The World Health Organization has, through it's called modeling, has predicted that by the year 2050, like Dr. Ndari said, there will be like 90% of deaths in Africa because of multi-drug resistance. And if we cannot have an alternative to these conventional antibiotics that we are using now, there will be a problem. Look at the burden. The burden is mostly in Africa, and the burden also in Asia. And yet in Africa, we don't have enough research to come up with alternative interventions for these multi-drug resistant drugs. So next. So that takes us to an era. Like a decade ago, we used these viruses called bacteriophages. These bacteriophages are viruses that infect and kill bacteria. They are very host specific. They target only specific receptors on a bacterial strain. So that virus is specific to a specific bacteria. And they are abundant in the environment. They are everywhere. They're the most abundant viruses that we have in the world. And they are able to inject, let's go to the next slide. If you look at the therapeutic concept, this is the structure of a bacteriophage. Biologist, this is the structure of a bacteriophage. It has a capsid, it has a genome, either DNA or RNA, and it injects a bacteria and injects its genomic material into that bacteria. It uses that bacteria as a machinery to produce more viruses, and it has produced more viruses, it bursts that bacteria out, and those viruses goes to another bacteria to inject, to cause that infection. So if you look at this micrograph above there, is a phage that of Acinetobacter baumannii, which is one of the multi-drug resistant bacteria also, that there was an infection here. The phage was able to infect that bacteria, and later it caused lysis. So we want to harness that concept so that we can use that concept in treating different bacteria that have become multi-drug resistant. Let's go to the next slide. So, in the lab I'm working, I've isolated bacteriophages for Pseudomonas aeruginosa. One of the bacteriophages I've isolated is called bunaviruses, others are called brunogeviruses, and they are ficmiviruses. These three viruses have a lytic activity. They kill that multi-drug resistant bacteria. But there's another unique bacteria called a fixivirus that I'm going to talk about today. It's a big virus. It's called a jumbo phage. If you compare normal viruses of Pseudomonas aeruginosa to this jumbo phage, is that this is a bigger phage that has quite elaborate lytic effect on these bacteria. But the problem we have, if we go to the next slide, there's something we call the viral dark matter. We cannot use a virus when we don't know exactly what it is. These fixiviruses, those jumbo phages against Pseudomonas aeruginosa, they have, like, if you do a sequencing and try to read their genes, most of the genes are not known what they are. And if I come and tell a patient that I want to treat you using a virus, but I don't know what's in that virus, then it becomes skeptical. I cannot use something I don't know what is in there. So it means the genome complexity of this virus is that there are like 80% of the proteins in that virus that are not known. And that's why we need what we call in silico computation, using various databases that are there to hypothesize the function of different proteins that are found in these viruses, so that we can convince people to take this as an alternative to antibiotics. So there are techniques that are there, like AlphaFold 3, which is an artificial intelligence that is used to model proteins. Interpro is a database that you can read motifs from there. And there's molecular docking. A lot of people are talking about docking here. And they also CAST-P to see active sites in that protein. If you go to the next slide, please. So the objective of this talk today is to investigate the function of one hypothetical protein in that virus, so that the virus was isolated from a wastewater in Nairobi, and I'm going to use this through in silico approach. So I designated that protein that's hypothetical like a sample of what should be done to all other hypothetical proteins that are in that virus. So if you go to the next slide. So the methodology. After isolating the virus from the environment, I go in the lab, I do purification. I do DNA extraction from the virus. I have the DNA. I do sequencing so that I can have the reads, the genome itself, and do bioinformatics, that genomic annotation, so that I can know exactly what's in that virus. And then I do hypothetical protein selection. Then after hypothetical protein selection, I'll do now physicochemical characterization, where I see solubility of that protein, of that hypothetical protein that I've selected. You see the thermostability of that protein. And then we go to domain prediction. The domain prediction will search through the databases and see motifs in that genome of the virus, and it will try to give the functions from that genome. Then later I'll do secondary structure prediction. I do the tertiary structure prediction. the active site prediction and do molecular docking so that I can infer some of these inferences. Let's go faster, please. So, in annotation, this is the Kisii virus. Now, the genome of the Kisii virus is a very big virus. For if you compare to other bacteriophages, it has a genome size of 277 base pairs. It has 70 RNAs, a unique feature for only this jumbo phage. It has total of 406 ORFs, coding sequences, but 318 are unknown. So that is the problem, the viral dark matter. So we want to try and infer these 318 coding sequences using just one example to show people that we can do more so that we can have a full information of this virus and then we can use it for therapy, because we cannot use something for therapy and we don't know what's happening. Next. So we start with hypothetical protein selection. So I selected a protein from that genome that is, in its genomic context, it's between the DNA polymerase and the viral protein and the viral structural protein. So that means this hypothetical protein could either have functions in DNA transcription, regulation, or it could be just a viral structure. And if I did what we call blasting in the NCBI database, and I found that most of the Kisii viruses that have been isolated from the world also have that protein, and in that protein they also still don't know the function. So if I find this, then I will update the database so that we know the functions of this hypothetical protein. Let's go next. So we started with the physicochemical properties of this hypothetical protein using a web server, and we found the isoelectric point is 5.2. That means it's acidic, and if it's acidic, it can bind nucleic acid. Given that we have the hypothesis that it's either used in DNA regulation or it's either used in structural proteins. So it has a gravity index, is the hydrophilicity or the hydrophobic nature of this protein. And if it's negative 0.34, then that means it's hydrophilic. So that means if it's found in the cell, in the bacterial, then it could be soluble to the cytoplasm. The instability index means if the stability index is below 40, it's stable. But if it's above 40, it's slightly unstable. That means this protein is unstable and could require post-translational modification. Then the aliphatic index is the thermostability. It's at 82, it's stable. And it can be used experimentally. It can be used in vitro and in vivo. And it's stable in various systems, mammalian cells, yeast cells, and even E. coli cells. So the hypothesis from the physicochemical properties is that could be this hypothetical protein exhibits biochemical features that are consistent with the nucleic acid binding proteins. Next. So if we go to the functional domain, if you blast in the database from the different, the NCBI, CDD, the BFAM, these are the domains that were found there. And most of these domains were pointing this hypothetical protein to a transcriptional role of regulation, to binding role. This is C2H2 type zinc finger there is for DNA binding, transcriptional role, and also for DNA repair. So we continue and say that this protein could be, it modulates host gene expression and can also bind nucleic acids. Let's continue. So in secondary and tertiary structures, this is where we use artificial intelligence to model. By having the amino acid arrangement, the artificial intelligence can model the folds of that protein. And you cannot use artificial intelligence without validation. So after you model the folds, we have the secondary structures that shows that it has 44.24 alpha helices %. That is 5.55% beta strands and 50.21 random coils. These are random coils. And if they are random coils, that means this protein is quite flexible. And if it's quite flexible, then it's suitable to the dynamism of binding. Yes. In the 3 dimension, at the residuals between 130 and 140, where we said that it was C2H2 zinc finger, we had a PLDDT of greater than 90. That means the prediction confidence is high. That means if the prediction confidence is high for this AI, that means this protein could be also, it could be used in DNA binding or nucleic binding during the phage infection. Let's go to the next slide. And so we used what you call Ramachandran plots to validate that prediction. Ramachandran plots uses the atoms it uses the atoms that are in the amino acids. So at these red parts, these are the core regions. That means if the residues are in the red part, the accuracy of the eye was quite well. The yellow parts are the allowed regions. So they're not quite accurate, but they are allowed. The green parts are the generous regions, but they are disallowed regions. And if you see, look at these proteins, there were only like these two disallowed regions. So if in a Ramachandran plot, if the residuals in the core regions are more than 90%, then the prediction of that AI tool was quite well. So we validate our 3D structure that it was predicted quite well. Let's continue. So we must now find the protein's active site, and we use a tool called CAST-P. This is computed atlas of surface topography of proteins. And this will give us the place where we will go and dock our ligand of interest. Because we have that hypothesis that we are using, could be this protein is a DNA-like binding protein, we can use a ligand that resembles that the DNA purine salt pyrimidines. Yes. So the CAST-P will give us the active site. Let's continue. And now we do the molecular docking. So I chose pacimidazole because it resembles the purine. Yeah, the purine is part that makes the adenine and the guanine in the DNA. So the pacimidazole was selected as a docking ligand due to its structural resemblance to the purine and its potential to form hydrogen bonds with the DNA binding domains, because we predicted the domain as the zinc fingers and transcription regulatory pockets. So this is the ligand. This is one of the derivatives of pacimidazole, and the target is our predicted protein, and the active site that we found using CAST-P is the site where we are going to dock, and this is the results. And if you look at the molecular docking results, the affinity, you could find at mode number 1, the root mean square deviation was 0. It means there was perfect binding. Yes, that means the protein has confirmed that it's used in binding nucleic acids. And there are also like 9 modes of binding. There are like multiple binding. That means it could bind more other ligands. It has multiplicity, multifunction. So let's move next. So, of course, the limitation in using computational tools, we require experimental validation, and in this case, we have to do co-immunoprecipitations. That's done in microbiology lab and immunology lab, and use X-ray crystallography and cryo-electron microscopy. And the docking, we use only one ligand. We can use more other ligands that can resemble pyrimidines and all other those. And then in future, because we were talking about nanotechnology, and because I'm also working in a lab where we are trying to come up with this alternative medicine, I was thinking of modeling an endolysin protein that is used, the key lytic protein in bacteriophages, and model it, and then maybe synthesize it, and then use nanotechnology to deliver to these bacteria. Because I've taken a lot of time, let's continue. And I'm working at the Kemri Rare Lab in Kericho, and I want to thank you. Maybe if you have questions. Sorry for taking time.
Give him a clap. Thank you so much for that nice presentation. I think I want to open the session to allow questions. But as people are thinking on what to ask, I want to ask as a simple question, but can be a little bit complex. What you sequenced, what you isolated, was it RNA or DNA?
DNA.
DNA. And you said they have RNA, right?
The phage could have either DNA or RNA, but most of the phages, they have DNA.
Okay.
Yes.
Because my thinking was that if maybe they had RNA, and we know RNA is actually not stable, it continues, changing its genetic makeup, then we could be able to actually have a problem. But then how do you ensure that there's that genetic stability of what you isolated? Because if you're going to culture it today and you want to subculture it the next day and use it, chances are high that mutations will actually take place. And that means you may not be able to have the similar proteins. So how do you ensure that what you are actually getting is consistent for it to be applied? And then another stupid question, suggestion is, this is not a stupid suggestion, but it's a good one. You can also be able to consider the proteins that you have already isolated, subject them to antimicrobial peptides, right? They can actually be able to interact, and that way you may actually realize that whatever may be the proteins you are getting from there, they actually have some other antimicrobial activity, and I think that can also go a long way to help you in terms of what you are doing currently.
Sure, I've not
I'm
isolated
forgetting
proteins.
a third one, but it's coming. Hey, more questions?
I've not isolated proteins. Maybe this is just computational, just reading, just bioinformatics. But the next future is to isolate proteins.
Okay.
So I've not yet isolated them.
Why I'm saying that is you said you had this one hypothetical protein. That is how I picked that protein.
Oh.
Yes.
Okay. The hypothetical protein is from if the genome that I have, when you read it, because we cannot, if I have a virus and I have the genome, is bioinformatics is I'm reading from that genome. And if I blast it all over the world that have isolated these viruses, this particular protein does not have a function. So I'm trying to infer the function.
And maybe where you were identifying these functions from, in quotes.
Yes.
Were you using the normal protein data bank, or the bacteriophages have their own bank against which you subjected them?
I use the normal protein data bank. Yes.
Then that could be, anyway, I think we have a discussion. Yeah.
Yeah, thank you so much. My question was in line with what Dr. Odari was talking about. And what I wanted to find out was this. In terms of the protein that you've selected, and also the issue that you've said that there's about 318 which are unknown.
Yes.
So the first question would be, is it an issue of mutation as to why they generate the 318, or it is inherent on the phage itself, on the bacteriophage itself, or maybe any other issue? That's one. The second question would have been in terms of isolation, which you said you're moving to. I would want to see if you're able to isolate even a protein of some sort. Then you do protein crystallography on it. I don't know if you can do the protein, or you can do the RNA, or you can do whichever part of it that you can do, so that we can actually look at the structure.
Okay. For protein production, in biology we use mostly E. coli, where if you have the DNA sequence and if you have the model protein, you can, like the way insulin is being produced, yes, with recombinant technology, yes, you just give it the reads, and then you use the 3D structure as your validation in the final product, yes. And about the 318, I'm using this one hypothetical protein as an example so that we can read out, we can predict all the functions from the 318, and we can be able to convince, like for example in Kenya, those who regulated the pharmacy so that they can allow us to use these viruses for therapy. Yes, because they are very skeptical that there's a virus that is trying to treat, but you are not giving them the full information. That's why this virus is not yet used for therapy. And it's been proven that it's able to eradicate this multi-drug resistant bacteria. Yes.
Yes, your presentation is a very good one. And on my side, when you talk of resistance, it's associated with the bacteria.
Yes.
Forming an out covering which is impermeable to antibiotics.
Yes.
So now, when you introduce this bacteria of your interest, which substance does it produce which makes this membrane permeable, so that now the antibiotics can be able either to go in and destroy either the mitochondria, the ribosomes, like that. So which chemical does that bacteria you are producing, which breaks that impermeability?
I'm talking about a bacteriophage is a virus. It's not a bacteria. And like HIV will attack your CD4 cells, it will like the same. The virus will attack the bacteria. The virus, you know, when it's out, it's death. When it's in, it's alive. So when it has compatible receptors on the bacteria, it will attach itself on the bacteria. And then it will inject its DNA into that bacteria. It will use that bacteria as a machinery to replicate its DNA and form more proteins of that virus. And then it will produce more virions, and then it will burst the cell. And then it will go and affect other bacteria. So that is the principle that we are harnessing in therapy. So it's not like a chemical, but it uses an enzyme called endolysin. It uses an enzyme called holins, and it also uses an enzyme called spannins to act on the peptidoglycan layer of the cell wall of a bacteria. Yes, I think I'm clear.
Oh.
Yes. Yes. Yes.
Sorry to take you back again. If I did get him right, I think what he was actually asking is the fact that bacteriophages now seem to be an alternative to the multi-drug resistance. What could possibly be responsible for the lysis or that actually kills the bacteria? I think that is what he was asking. I don't know whether I'm wrong. Yes. That's
If
what
you
he was
look at the
asking.
anatomy of a bacteria, I know you're a biologist, we have the peptidoglycan layer, the cell wall, and the cell membrane. The bacteriophage has an enzyme called the endolysin that acts on the peptidoglycan layer. It hydrolyzes the peptidoglycan layer. It has an holin that forms pores on the cell wall. It has a spannin that forms spores on the outer cell membrane for gram-negative cells.
So then the answer should be like it has several ways on how it works on
Yes,
the bacteria
yes,
to actually kill
sure,
it.
sure.
Simple as that. Thank you.
Thank you.
Any other burning question?
Yes. I think.
Okay. All right.
Okay, so we'll have our last presentation, unfortunately. So that, thank you so much.
I just wanted something.
Sorry. Yes.
A layman question, eh?
Yes.
Okay, Meshach, you have done very well. It seems you have a grasp of your area. Me, I'm just asking as a layman because I'm not a biologist. We have these antibiotics which are normally sold. I don't know whether they are antibiotics, things like ginger, saumu, I don't know. Do people also develop when you are maybe from your data? Did you also sample the same, or you are just using these conventional medicines? Because I'm made to believe that also all the others are also antibiotics. I don't know.
Not done work on the gingers and those stuff. I've only done work on these viruses and those antibiotics, so I cannot answer it authoritatively like I could.
Okay.
Sure.
Okay, thank you so much. The last presenter, can come on board. I think they'll use a different system there. So Cynthia.
Okay, good afternoon, everyone. I'm Dr. Cynthia from Mathematics, and what I'm presenting is on an application that we are developing to aid in supply chain monitoring, as well as post-harvest losses, and also to incorporate an AI food scanner to be able to detect any toxins in the foods, as well as detect any let's say unnatural or unrequired moisture levels. So because of interest of time, I'll be very fast. We have already done the stakeholders engagement. We were able to gain very good insights from that, those which are actually helping us know what to incorporate in this application. So I'll go through that very briefly. As you can see, this is what we're able to get from the elderly people. These are going to be the consumers of our application. So that is based on gender and the frequency that they buy. This will help us know how strong we need our application that is actually based on AI to be working. We need to know the strength that we need to embed on that platform. So we can move on to the next slides. This is still on the stakeholder engagement. That is the elderly. They are one of our stakeholders. Then the market source, where do they get their vegetables as well as cereals now, whether they use mobile phones. Of course, if they are going to use this application, they need to have at least a mobile phone. But we also, from our stakeholders engagement, we were able to realize that they would also prefer offline mode. So that is something that we are actually working on to incorporate in that. Then some of the challenges that they face when they are getting their foods from the market and the delivery methods that they do. So you can move on to the next slide. Still on them, we were able to check on the important factors for the AI digital market that we are developing. They were very keen on fresh produce as well as nutrition, affordable prices, reliable delivery, and ability to pay using mobile phones. So that is something that we have been able to actually embed in our program. They be able to pay from home. They be able also to be able to check on their food quality. That's why we are having the food quality app. Then we're able to check whether these aspects or these factors are significant. Because of financial constraint, we had a very small sample size that was 30, so this made it affect some of the factors being significant, but the direction of our coefficients was able to let us know the interaction effect of these variables with AI adoption. And as you can see there, males have a positive coefficient. The most elderly people have a negative coefficient. Of course, these people are not maybe open to adopting AI tools and that. Then you can see the trust in AI, how it will affect AI adoption, the internet, the weekly use, and also the cereals buying monthly. We can move on to the next slide. We also have the riders. These are the people who are going to be delivering the produce to our elderly people and our disabled. So we also are able to engage them. And these are some of the things that we were able to get. Of course, one is the distribution of them in terms of ages. You can see most of the riders that we're able to engage are quite young, so that actually increases their level of AI adopting that application, which is actually AI driven. Then we're also able to check what time do they prefer, so that we can make sure that we actually recommend that even to our buyers, that this is the time you're most probable to be able to get a rider. So that is the time maybe you can be able to order your product most preferably, but of course you can order any time of the day. Then we also engage cereal sellers. Of course you cannot eat vegetables alone. You need to get some cereals. So we also engage some cereal sellers so that we can be able to embed in our application. Then we were able to check on their digital adoption trends in terms of gender. We were very keen to ensure that we observe the GEDI rules. So you can see digital adoption in terms of the sellers. Men were actually higher than women, as well as the digital adoption in terms of now the consumers. Also male were also higher than females. And also here there are some of the features that they recommended that they would like to have in our application, that multilingual interface. So we are considering Swahili as well as English. Then ease of use, we're making sure that we're using very, very easy interfaces so that they're able to navigate through the pages. Then this is just basically those insights that I was able to talk to, include AI scanner, integrate the GPS and that. So I think you can be able to share what we have been able to develop so far. Then I can be able to go through that also very briefly. Yeah, so this is the application as it is now. You can see the page that is being displayed there. This is our digital market, which is actually AI driven. You can see it has a chatbot. You can actually just scroll up. Yeah, you can see we have the chatbot section there where you can be able to interact with the chatbot in case you're not actually able to navigate through the products on your own. You can be able to fill out the products in terms of maybe what you want to buy. Of course, in the future, we hope to, if we get more funding, we can be able to expand it even to more products. You can go back to the landing page. You can go back to the landing page. You can see we have actually also embedded the payment methods. Then you can go back to the landing page. You can use the back button. Yes, that is, you can see that is our landing page. So that is just basically the display. But you can see we also have now the other tools there. That was the digital marketplace. You can click on the AI for food quality insights. That is actually where we are now specifically now integrating with the chemist. So in that, the AI food quality section, then we will need to check nutrition levels of the foods, toxin levels, as well as abnormal moisture content. So we have actually engaged some chemists so that they can be able to give us data on what are the required toxin levels in not toxin levels, but what are the abnormal toxin levels when we, in terms of the cereals, as well as the abnormal moisture contents in terms of the African leafy vegetables or any other vegetables. Then we will be able to use now that data that we get to train our AI tool, such that when you use now that AI food quality scanner, you're able to know whether your cereals have exceeding toxin levels or your vegetables have exceeding or abnormal moisture contents. And through that, we are actually going to be able to prevent these diseases such as cancer, which are most of the time caused by absorbing toxins. We know that some of these cereals that we consume, they have high levels of toxins, and that's actually cause of cancer. So we are really focusing on prevention rather than now the curative sector by developing this AI app tool that is going to help us to be able to determine whether our food is fit for consumption. Once you determine whether your food is fit for consumption, then you can be able to sell it to your consumer, and the consumer will also be able to access this food quality app so they can be able to check. We know some of our sellers, they're very cheeky. Anaweza eka hapo yenye ni mzuri, then they sent you what is not actually fit. So you will also be able to check in your end as the consumer whether what you ordered is what you got. Thank you. That is my presentation for today. Any questions?
You've talked about the safety, which is actually very good.
Okay.
And I want to commend you for the nice work you guys are doing. And it's good you are actually acknowledging that some of our customers are not faithful, I can say, and they can actually end up sending food that is not value. But then the question is, you know, you have given this boda 2 kgs of nuts to bring to my house, right? Yes. How do you scan from your end to actually know that the food which was safe is the one that has been delivered? That's one question.
Okay.
Then a second one is, perhaps you've said you're going to use an AI scanner, right? How do I know, well, maybe this is a suggestion. You need to look at all the components that maybe can affect people apart from aflatoxins. That's just but one. Because I can equally maybe buy spinach which is contaminated with E. coli, right? And that is actually going to be poisonous into me. So you need to consider a number of factors that actually define what we call as safety. For quality, you talked about the nutritional elements. I think that is okay, and that may not really be able to change. But when it comes to safety, there is a question mark there. Like, for example, I don't understand how you are going to have an AI scanner telling me that the genetics you got has maybe this amount. Okay. And vis-a-vis what has been...
Yeah, maybe I can take your question.
Yes, thank you.
I think the first one is how are you able to check whether what the rider was given is what you receive? Yeah, so the first thing that we're going to be doing is we are going to, this is of course the back end of development of the app. Once we develop it, then we are creating a mobile app. So you'll be able to install that mobile app from either Android or Apple Store. So once you have it, it will have those functionalities, the digital market as well as the AI food safety scanner. So you as the consumer, you'll be able to access that. And once you get the food, that's why we are saying we are going to train our AI. You know, once you get the AI model, you can train it to what you want. So if we feed it with what are these and required levels of toxins, we are not actually focusing on aflatoxin alone, even we are focusing on other toxins, even in the leafy vegetables. So that's why even the skuma wicks, the way you're saying, it's not really aflatoxin alone. That's why we're focusing on vegetables as well as cereals. And as you can see, cereals, we are not having maize alone. We are having the green grams and others. So we're actually getting all that broad aspect of it, yeah.
Thank you. Another question? Right. Are you going to have all the barcodes for every food?
No, once we train that AI food scanner, then it will be able to detect what image is this. If it's maize, it goes directly to the back end of maize. Yeah.
Okay. You have also presented very well. I never knew that you were a programmer also. Mine just a layman's question. Yes, once I make an order, because I've seen some cereals there, something. Yes. Does that order also link to, do you have a database of those border riders maybe who have registered to your app so that the seller and the transporter are also linked to the system so that I know maybe if I make an order, a certain rider will just bring my...
Yes,
product.
actually, you see, we have some selected riders who we are doing in this trial phase of the application. So once you order, then it will link you to the seller. Then now the seller will be able now to be linked to a rider, a specific rider. And we are using also GPS technology. So we will be able to link you to your nearest market and also link you to your nearest rider in that market. But as for now, we're just confined in Kisii because we're just during the trial phase. But that's how we are developing it. So if now you're ordering maybe from Nairobi, let's say, for example, when we scale it up, then it will be able to link you to the nearest market. That's where you're going to buy from, and also the nearest rider who is actually using our application. So that you'll be able to get it at your doorstep.
Okay.
Yes.
Yeah, I just have one. So you talked about food quality. And you said moisture content. Are you looking into ways of get the experts, guys who do food quality, guys who have kits that can sample other, there is ways to tell that there is mold around. So are you making headways into getting these people on board so that they can actually be randoms, checkers for you?
Yes. Actually, we are not working in isolation. We are working with also agronomists who actually know these foods better than us, mathematicians. And we're also working with scientists who can be actually people from chemistry who can be able to tell us these toxin levels and what is required. And what actually, we also have a cold room, which we're also developing an AI app to be able to store now these African leafy vegetables or the vegetables. So now we will be able to get an AI technology which can be able to regulate these temperatures in the cold room. then they will also tell us which are the optimum temperatures that will make this spinach of yours not go bad or grow molds. So we are actually working with agronomists on that. Any other question?
So IT, I promised, I hope you're not folding up. I promised it. I'm going to entertain everybody with that while we wait for the awards session. It's only 2 slides. So you just go to the very back. It will be under Nixon Mwebi. Sure, you got it. Thank you. I'm glad you're ahead of the game. Yes, just I am doing that because I've got a lot of questions from students and faculty, but now I'm just looking at faculty. I think we have very few students, if any. So if you're doing your PhD, you're really almost faculty, so it will follow under this. So there is this notion, and please forgive my French and my assumption, but all that I'm saying here comes from the very fact that I too sat where you are. I got my first degree from Kenyatta University, so I was at that level where I also, in my case, I actually wanted to leave the country before I did my first degree, and it was very interesting. I really didn't buy into the program that I was doing, so I went in and I told my family, especially my parents, I told them, I really want to go out. And they looked at me, they were like, are you out of your mind? You already have an admission to the university. What's your problem? And I said, I'm not very excited about the program. And they said, no, you got to do it first. So, you know, I was very young. Do you understand being young? Yeah, I was very young, and I really wanted to sabotage the process. So I went in that first year and I wanted to fail, because that time if you failed, you got what? This code. I mean, it's amazing. I was told yesterday that the student can fail and do it and fail and do it and until they pass it, and I was like, that's not fair. I mean, we suffered. You fail, you get a sub 1 and you're out. You fail 1 and you're out. So anyway, I went in to fail that first year or whatever it was, and I didn't fail. But I didn't do well. You know, mediocre, you understand mediocre? Is the worst place to be. So I decided, okay, I might as well just pass and do this thing. So then after doing that, I actually tried to leave the country, and it took 4 years to do what I wanted to do. And Britain, I applied to Britain for 3 years in a row. And I applied to Cambridge and Liverpool. I didn't apply to too many places. Liverpool always gave me admission to the environmental chemistry program, but they wanted $250,000, you know, Kenya shillings that time. That's almost like what? Think about it in the 90s. What's $250,000, Mr. Nyancha? What is that like? Yeah, it's around $1 million or $1 million plus. You couldn't come across that money now. That's the time you are earning about 5,000 and somebody's asking for $250,000. What are you talking about? So it was an asymptote. And then I applied to the US and I had the wrong information. Here is how I had the wrong information. And there's a reason I'm giving you this background, actually, for this information. Information is power. So I had the wrong information. The time I was applying to Britain, I was also applying to the US. And I had a lot of people in the US. But you know what? A lot of the people who came from where I hail from, they went to the US not with the mind of studying, but with the mind of making money. Do you know anybody like that? So what they did, they went to the US. We have what we call 2-year colleges. They are community colleges. They're very important over there. But they did this, and you can enroll. That time you didn't have to be full-time. So long as you have a class or two going on, you can do whatever else you want to do. So the people were busy working, doing a class here and what? There. They are the people, me as a graduate already, I'm asking to tell me which schools to do what? To apply. And I'm getting information from these 2-year colleges. I'm sending them my transcripts, and they are telling me, you've done a 3-year program. We have a 4-year in America. There is no way. You got to go back and do 1 more year. Where do you go back to do 1 more year when it's a 3-year thing? So you see, and for 3 years, I was in this cocoon, or 4 years, where I really didn't get the right information. And then, fast forward, it's a summer of August. I'm walking down the streets of Nairobi. I meet one of my former lecturers. And he looks at me and says, Nixon, you're still in the country? I said, of course I am still in the country. He says, but for the last 4 years, I've given you recommendations. Out there, by the way, recommendations are very, very important. Very, very important if you're going to do a research program. Because they want to know what kind of a person you are. It's almost like a marriage relationship. If you suck, you have no discipline, I don't care, you have A's, I'm not taking you. So he said, well, wait a minute, I've been recommending you, what happened? I said, I'm always getting these admissions, but I don't have the money. And then he told me, come to my office on Monday. And I've just, 2 days ago, I came from the US, and there's this institution that I think will take you in if you do what? If you apply, because one of the guys who is chairing that department is actually from Tanzania. I think so, he said. So I said, I'm so tired. I've done it for 4 years. You know what I mean? I mean, you kind of get tired, do you? Yeah, but something, I guess, providential told me, just go. Cut the long story short, I went. I got this. That's all he gave me. Just a piece of paper with the name and whatever. And I Googled it. I found the department. I found whatever. I sent in my application, just as I always do. And guess what? One shot, I got an admission. And that admission letter, you got to be very careful. It came to my workplace, and somebody somewhere just misplaced it. So I wrote them back. I said, hey, now that was in August. I wrote them back in November, December. Said, no, I think it was, yeah. And I said, what happened? You guys never wrote to me. The assistant dean wrote back to me somewhere in early, and I got that letter in March. He said, we actually gave you what? Admission. We wondered why you didn't come. But so, and where was I? Okay, I'm back on. Thank you. You know, we are useless without you guys. Thank you so much, IT. So he wrote back and said, if you wait for the fall semester, we'll not just give you admission and a little bit of support. We'll give you 80%, 90% what? Support. So you realize that's what happened. So that was I'm getting this letter in March. I prepared myself. I went in. This was the fall of 96, and the rest is history. Got my master's, got my PhD, and I'm here. Why am I telling you that? So what did I get? Just what? Information, right? He just gave me the right information. Just the contact, and it did everything. I could have, by the time I had friends and relatives over there, they didn't pray a lot in doing that. In fact, they were the ones who gave me the wrong information. So I'm telling you all this to tell you that what you need is the right information, not even a friend in the US. That doesn't mean I'm not willing to help you navigate, filter, and do. But I want you underlying to know that information and the right information is what you need. So there are many ways that a faculty can actually take advantage of those opportunities. These are the platforms. The first one is postdoc. Why did I put that as number one? Sitting where you are, if you have a PhD, all you need to do is scout. Scout means scan the programs in the US and find somebody who is doing the phages or who is doing the natural products and all that. Excite them the way you excited us about those two molecules. And they'll be like, there's this guy. I mean, he's in Kenya. I don't know about it. But you'll start having conversation. And as you start having conversation, the postdoc, all it does is that this guy has an NSF, National Science Foundation grant. He has graduate students. But now you already have a PhD and you know how to do research. So what did they do? They take you into their lab. You stay there for 6 months or 9 months or 1 year, and it's beneficial to them and to you because you go in, they don't have to train you. You already know how to do what? Research. You just get the equipment. They give you some stipend to live on, and because you're coming from Kenya, the exchange really will be good for you anyway, right? So you stay there for 6 months or 9 months, and after you do that, you get some publications and some exposure, and guess what? The school next door is needing a faculty to teach this thing, and guess who is applying? It's you. And now, before you know it, that little postdoc that you did for 6 months puts you as a faculty in the next school. And then people start saying, so you know this guy and Dr. Mwebi were friends. So Dr. Mwebi told him, no, nothing. You just scouted, applied, and you got it. Postdoc, there is no deadline. There is no right time or wrong time. Anytime you can apply for a postdoc. It just depends on whether that guy and what you want to do. Most of them will be, if they are in research and intensive research, they will be in what we call R1 universities, research intensive universities. So these are the big dogs in any state. You all know what I'm talking about. We have, in the US, we have universities that are for intensive research. There are some that are, we call them like master's granting institutions, and we have undergrad. So when you want to really take advantage of that, you want to go to the what? Research intensive, and you'll do that. Fulbright exchange for people in the teaching as faculty like we are, right? The Fulbright is like our Nobel laureate, isn't it? It's like if you get a Fulbright to go anywhere, it's like you're really at the cream of it. Because what is it? They give you the Fulbright exchange program. Will take, like I came on the Carnegie diaspora. The Fulbright will bring you for somebody from there for a year to teach here or do research. And it will, by the way, do you know that it will take somebody from here to the US to also do the same thing? Did you know that? Yeah. So the Fulbright, the Carnegie may not do the exchange backwards yet, but the Fulbright will bring you from there to here, or bring somebody from here to where? To there. So what I can do for you is to find you the links for the Fulbright program. I just made an assumption that you already know. So without even so what do you need then when you are doing the Fulbright? You need, if you are a biology guy in the area of biomedical, again you look for guys who are doing that thing. And you tell them, look, I want to come in on a Fulbright. Most people are not going to refuse, especially if you work in that area. Why would they not refuse? The university there is not going to pay. The Fulbright program pays you an equivalent salary to, if you're an assistant professor, they'll pay you that. If you're an associate, they'll pay you that. In other words, if I came on a Fulbright, the Fulbright program will pay me my full professor's salary right here for 1 year. Kisii University doesn't have to spend a penny. So why would the guy refuse? Do you see what I'm talking about? The same case on the other side. So these platforms are wonderful. The only reason we don't take advantage of them is maybe we don't know about them, or we are hesitant, or we think we are not good enough. I mean, the worst they can do is to say no, right? And if they say no, are you dead? You're still a professor. Come on. I mean, actually, I applied for the Fulbright before it was 2 years in a row before I went to be chair in California, and I was serious on it. But when I went to be chair, I got too busy. I was a finalist twice, 2 years in a row. What it means is that a finalist, well, I was a finalist in the US, and then it comes to the region. What means is you are accepted, your program, your proposal is accepted. Then it's sent to the region that you want to go to. And I was adamant about wanting to come to Kenya. That's the time we had a maize problem, and we had that maize, you grow it and it was dying, and that was my proposal, to come and do this thing. But when it came to the sub-Saharan, the competition for Kenya, people want to come to this climate and the animals, the competition is too much. And so twice, and that's how. But that tells you that you can actually be a finalist, and you can actually do it. Faculty Scholar Exchange is similar to what I'm doing. But here is, you can actually discuss with the universities over there, and you can have an MOU, or you can just have a, like, for example, I'm chemistry and an analytical chemist, and we can have something, and if my university is willing and your university, then that's a local arrangement sometimes. And then research conference. I know sometimes you're like, where am I going to get a ticket? But sometimes you sacrifice. Send your abstract to a meeting, American Chemical Society, the ABRACAMS, which is for biology, or American whatever, biological whatever. Go for a meeting somewhere once. When you are there, you are not only just going to present, but you are actually interested in doing what? Networking. And from that, when you come back and you write to that guy, that's how you get to do this Fulbright or this exchange. So those are a good The next slide, please. Next slide. The last one, which just elaborates more on these things. Yeah. So there we go. Invite a scholar or a colleague from the US. Bring him as a program reviewer. Bring him as an external examiner. Bring him for a talk. It doesn't matter. Maybe they can afford the ticket. What do you care? The worst they can do is to say what? No. What do you lose? Nothing. Create a bridge. Send your student. Right? Yeah, good master student who can do things, encourage them to apply for their PhD in University of California, Berkeley, or University of Maryland, College Park, or wherever. When the student goes there, guess what is interesting? They are talking with that professor, and the student tells them, my advisor over there is really very nice. If you can have him for 6 months, you guys will do a lot of work here. He's speaking in the ear of the what? The investigator. You already have somebody in there advocating for you. Well, if that doesn't work, in about 3-4 years, your student gets a PhD, right? And becomes faculty down the street. Guess what the student is going to do? Invite you. So you are creating bridges, right? You are better off than where you were. That's how I see these things working. And then get involved by a scholar in the US. Again, Fulbright, postdoc, visiting professor. You can be a visiting professor there, and they can be a visiting professor here, right? Don't think you can't do it. Well, if you don't have stellar research, become a visiting professor in a teaching institution. You can teach actually, can't you? Yeah. So who tells you that you cannot get a job in the US just to teach for 1 year? We always have those. Like now, I came for a semester, and I had to have somebody cover me. If I was coming for a Fulbright for a whole year, who do you think is going to teach my classes? A visiting professor. And sometimes we don't care where they're coming from, right? If we can't find somebody locally who has the ability to work, then we'll poach them from wherever just for a year. So yeah, think about those things and you'll see them advertised. And so one of the things I can do if I come to it is, again, send you some of the links of the places where you'll see these things advertised. Research conferences, I've already talked about that. Send an abstract, network, initiate collaborations like the MOU. We are working on one between University of Lavan and what? Kisii University. It's in the works. And when that becomes real, what it does is that now there is already an avenue. We know what to do with you when you come, and the university here knows what to do with somebody who comes here. That is why it becomes easier. Now it's hard because what do? Invite a faculty. What happens when somebody comes to give a talk? There is one thing you don't understand. Whenever I'm coming to give a talk in a university, I'm not just giving the talk. Before the talk, let's say I come the whole day. The talk is at between 2 to 3. What do you think I'm doing from 9 to 2 o'clock? I go to various rooms and you set up meetings with Professor so and so and so and so. That is what you do. When you go to give a talk, that day you meet with the faculty, you talk to them. What do they do and what do you do? So what ends up happening? You figure there is somebody, you have common interests, right? So those are also one very good ways of actually doing this. So most of the time we take advantage of that. I invite people that I'm interested in knowing and knowing what they do. They come for a talk, but they spend the day looking around and doing that. Initiate grant ideas. There are some things, for example, environmental is borderless. You understand borderless, right? Because when it is produced here, or all the coal banned in China releases all those sulfates and all that all over the world. So those ideas, grants that have those kind of ideas, can uniquely be supported even by the NSF in some ways, because they would still want to know what's going on. CDC does a lot of work with the Kemri and all that here, because diseases have no boundaries. We have Ebola here, and before we know it, it's in the US. So in grant ideas so these are many ways and many platforms that you can use. And I hope that noise helps you somehow to understand really that you just need information. Is what I got, is what I can give you anytime, now and even later. And of course, what are some of the ways I can also help? For example, if you are applying to places and you want to know from me, I'm interested in this state, this institution, that institution, you can pick my brain. What do you think? And I'll tell you, that one is very cold. That one is very racially a problem. That one is this and that. Those are some of the insights I can give you, right? Oh, I'm getting this kind of offer. What does it mean? I can also translate that, right? Some of the things that I had another slide, but I don't have it here. It has, the last slide was to do with your CV. In academia, we don't care about resume, right? You guys know the difference between a resume and a CV, right? A resume can be 1 or 2 pages. This is for industry or corporate world where you want to look good. So you use the language and all that. In academia, yes, we want good language, but we are just interested in who are you and what is it you've done. And most of the time, chronological. Do you understand? So, but there are things that I see you all put in your CV that are a no for us. We don't need your picture. We don't need your age. We don't need your marriage. We don't need a lot of that jargon. Give us your education, your academic qualifications, your publications, your awards, the mentorship, right? The granting, right? Now you're going to say, Doctor, maybe you need to really give us the best way. Now we even have AI. Just go online and look at a sample, and they will give you a sample for chemistry, for biology. Please don't send your Kenyan CV to go look for the job over there. First of all, they don't know what to do with it because you have given them more information that will make them compromise your, what do you call it? The name, not even, there is information that can compromise your identity. They don't even want to open it. So don't do that for yourself. Just give them what they need. Am I speaking English? Yeah, so please, I sit in a place where we look at some of these and we get some of those resumes that have that. We don't know what to do with them. Because we don't want to be responsible. I don't want to be responsible for your personal information. Do you see what I mean? So when you send that to me, I would wish I didn't even open it. So please, whenever you are applying or whenever you're doing, these are some of the things that are very important. Just give them what they need and keep it very, very professional. Okay? Thank you so much. Any question for me on that, please? Question? They are welcome. Yes.
Things that I think my colleagues would want in particular is if they are hoping to collaborate, might not be if they are hoping to collaborate, but not collaborate, yes, but not necessarily with you. You know, one thing I've realized is that there's a power in somebody referring you. Meaning that if somebody from biology and then you go and talk to the professor because you've met the gentleman, you've met the particular lady, and they ask you to, you could, refer them to that person and say, you know what, I can actually refer this person because I have that. So I'm asking you, on behalf of my colleagues here, that if at any point they see that there's a person, particularly in your university, that they might look at their profile and think, you know what, this looks very interesting. I don't know if this is something that we might be able to work on. And I know you might not know the person physically or personally, but I've realized that if someone refers you and they work in the same institution, it carries some weight. So that's a request that I'm asking for you.
So what you are asking, in fact, is not even a burden for me. When I was coming here, the biology department, in fact, remember, and I'm going to take him to task, there is even a colleague in biology who sent me information and wanted it to circulate in the biology because they were going to, the colleague and the sister were going to be editors of a particular issue, and they wanted the biology from Kisii University. Anybody interested to jump in? It was in ecology, something. I'll look for it. So what am I saying? They were already excited with the fact that I was going to work on an MOU, and they would have some colleagues here that they can exchange with. So it will not be a problem, but I'm only realizing one thing, having been in Kisii University for a while. You guys don't have forums like this. So if we had done this when I first came here, by the time I'm leaving in 3 months, each one of you would have, because what I would have done, like we did for the New Yorker, right, Dr. Obare, I would have invited 2 people from biology, 2 people from math, 2 people from whatever, and now we would create a place where they already kind of know some of the names, they would see what you are presenting, and now I don't even have to tell them. Are you hearing me? You don't have these forums. You need to create these forums. It doesn't cost the world to do this. If the university doesn't want to give you water and food and all that, just do it anyway. Am I speaking to you? Then I will invite my folks to come and listen to you, and now they know what is it you do. The alternative is for me to tell them to go to the website and look at what you have put there, but sometimes it's not updated. Even mine is not updated. Do you see what I'm saying? So they are very willing. Okay? I will say it again like I said it when I first started. We have some things we have that you don't have. You do have some things that we don't have. We have a lot of equipment, for example, in chemistry, that is used maybe 40 to 50%. Half the time it's available. Why? In the Department of Chemistry, we don't have a graduate school yet. We actually are well equipped to have a PhD program. But the coining of programs and the growing of programs in the US is much more regulated than whatever than here. You can't just come up with it. But you do have those graduate students. And they finish their coursework and they don't have equipment. And they can easily come and use our equipment for 6 months, 9 months. I'm sure biology the same. So they were all very excited when I told them that you guys have these graduate students who could possibly come, right? But now, even before the coming happens, there needs to be that exchange between faculty so that, yes, I can work with you on this thing, right? You can't just send a student and you don't know who they're going to work with. Are you with me? So I'm a willing party, just no problem. Another question. Yeah. Should I give you this? Okay. Postdocs, yes.
Postdoc within a range of 3 years. So maybe that I think has been a major challenge for me. And again, when it comes to writing grants, like for example, there's a time we did a Delta towards a grant. Mm-hmm. And the regret we got was that we don't have the experience to handle the amount of money we were actually applying for, 250 million.
That is true.
Yeah, so I think it becomes very hard, especially when we do grants. It becomes hard really to get somebody from that end that, you can work with. And I think, those guys are like, we have to build trust first before we can be able to do something together. So I want to believe that the way you've actually opened our eyes today, that we need to connect is very important. such that maybe before that thing happens, I think then it's a doable thing. But then generally, what could be the wrong thing with, 3 years?
Okay, so here is something about, and sorry that it's taking time, what we are about done, believe me. So here is the thing about this. The 3 years, sitting there as a professor, he's looking at the fact that I'm bringing in this guy, right, who's been professor for what, 8 years? So he's thinking that you are at the level where he can't be able to, handle, manage, do, your expectations may be higher. So this person who is 2-3 years is almost like a graduate student. They don't have very high expectations. They can work together. So it's one thing to tell a student do this, is another to tell a colleague do this. So that does come with some drawbacks. So that is where that breach becomes important. So you make a valid point. And when it comes to the monies, don't blame them for not trusting. You know what we do when we get money, right? So that's, we get good grants. And then, but there's one thing that we do that they don't do. Don't think there is no graft over there. But nobody sings about it. But let me also tell you 2 things that Western world has done with corruption. Number 1, they have legalized it. Legalized it. Number 2, they don't talk about it the way we talk about ours. Are you with me? Yeah. So your brother screws up at home and you sing about it that the whole village already knows. So who is going to come to your home now? Who is coming? Are you with me? That's number 1. Number 2, they legalize the corruption. Let me tell you this. If you are applying for a passport in the US, it's probably going to take 6 weeks to come. But if you want it in a hurry, you want it in 2 weeks, you just pay $500 more. Are you here? To the same government, there is no back door. The passport, you can get it for $125. Straight 6 weeks. But you want it in 2 weeks, pay $500 more, you get it. So they have legalized the bribing. And so if we do that, we are okay. So that's the difference here. And that is why they don't. So for them, when they come here, and believe me, I've had friends from Canada and US who wants to come and invest and do business and do things and whatever. Like when the oil was discovered, some of them came. And they go to these boardrooms, and our folks higher up tell them, well, you know how we do business here. Do I need to elaborate? Okay. So, any other question? So we do this to ourselves. Please. I know, don't kill me. Kill the people asking questions. He's looking, so he's gonna work on you.
Okay, thank you, Prof, for that very good information. Yeah, I also admit that the information which normally get from the people outside there, because most of us have been maybe friends or relatives there, and most of them are just truck drivers or nurses.
Thank you.
So there's no way they can advise you on matters academia.
Yes.
So this is one question which I may want to ask. What about if someone wants to teach? I was told that there's a way they convert the papers. Maybe if someone has a PhD here and wants to join the American system, must it be converted into the local? Because I have even a colleague who went there and is now a kindergarten teacher with a PhD. So I don't know how it goes about. Is it a must that it be converted or what?
Please. Again, you have gotten the wrong information. I'm glad you brought it out. So there is something that happens here, and let me say it, and you have been to the US and you understand what happens in some circles. And I'm going to be very transparent. Okay, don't quote me. I have nothing against people in places and all that. Engineers have left here and gone to become nurses, right? The one thing that happens is that I told you it's the mindset you go with from here. Now I'll give you some statistics that are just from my head because of what I hear and what I see. For every 100, I could say even Kisiis, who live here with a green card, mark my words, they live here with a green card. to the US, for every 100 Kisiis who live here with a green card to the US, probably only 8 to 10 are going to go do school, meaningful school, have a bachelor's and a master's or a PhD. Why? Because when you go with a green card, you have a ticket to work, do whatever you have to do. You don't need to have any status. You can do what you want. And in the US, you work for maybe 2, 3 months, 4 months. You can buy a ramshackle car. You can rent a house. You can be independent after 5, 6 months. And you're just as good as the guy next door. So then why should you go to school and struggle? And you can send some money home, and boy, they are building a 5-story building for you. You've made it. If that's what your mindset was. But when you go as a student like I did, you're on F1, you got to maintain your status. It's very expensive. You get peanuts when you are getting a stipend. See, the way you go, you become an assistant. You get an assistantship or a research assistantship for the 5 years you're doing your PhD. It's like a tutorial fellow. They waive your tuition, so you don't pay tuition or fees as you call it, for your study, and they give you a little stipend, money to do what? To live on, and they're very careful. If it's Kisii, they know that to have a 1-bedroom you need 15,000, and then to eat you need another 15,000, so they will give you a stipend of about 40. Just enough to survive. You understand? Because they don't want to have, if they give you too much, you will not be studying. You see what I mean? So most people will be like, come on, why am I going to do that when I don't have to be a student? So they can make, when you are making 40, the person who is working out there, we are making 40,000 a month. The one working out there, doesn't matter what job they're working, they'll make like 200,000. So you have to be very motivated to do that. So now the question you asked was, this guy left with a PhD and became a kindergarten teacher. 2 reasons, or 3. 1, if they were financially motivated, they could do that. In fact, I know of high school principals, and I was a college professor that time when I was beginning my salary. The principals or the headmasters were making double my salary as a PhD guy. Are you getting it? So it's not the question. You could be paid much higher doing something that is not what you want to do. Of course, the professor will come and overtake you, but at the beginning of it, he's down there, you are here, because of the level of work you're handling. Number 1. Number 2, you do have to translate your transcripts to the American system, but you can do it sitting here tomorrow. There are agencies, and again, please, Dr. Ogutu, remind me to supply this information. I can provide you links that will do that. So there are agencies that take you, here we do first class, second upper, second lower, and what? Pass. We don't do that in the US. We do GPA. So the GPA will be mostly 4.0 is the best. So you can have 4.0, 3.7, 3.5, 3.0, 2.0. And so when you are below 2, you really suck, right? So they will take your transcript that is a first class, translate that to GPA, and realize, okay, you are like 4.0 out of 4.0. Or you are like 3.7 out of 4.0. Like that. That you do. At a small fee, you send it to those people, they will translate it, they'll send it to the school you want, and they'll send you a copy. So you will know, even though I have a second upper or second lower, I'm like this over this. So when they tell you about GPA, you are not ignorant. So that does happen, but that's very simple. The people who have to go through fire, if you so will, are the people like medical doctors and pharmacists and people in that profession are dentists. So if they want to practice, you want to practice as a doctor in America or a dentist, that can take even 6-7 years trying to get into the system. You have to do the board exam. You have to find a place that is going to give you residency. That takes a minute. But you have a PhD and you want to teach in the university over there, well, if they didn't take you on a postdoc, you have a way of going with a green card or any other way that is legal. Go and attach yourself to the local university. Be an adjunct. We call them adjuncts, not part-time. We call them adjuncts. So I always, every other year I'm employing 3, 4, 5 adjuncts. Very easy, because I need them. Because somebody was teaching those 3 classes, like right now, I need to go because there are 2-3 classes that I need to be covered. I need to figure out who is covering it. Somebody was hiring somebody. I hope they will sign up and cover the class. And they will cover it for a semester or 2 and they go somewhere else because they got a better opportunity. So then I find you and you have this. You just come and teach for me for a semester or 2. But now you have experience. Now you have something on your CV and then you apply to the next program. So it really, you don't have to teach kindergarten. All right. I know I went so far to answer that question, but I wanted to supply some information. Please, are we done? So those guys can celebrate. So now, I think Dr. Gutu, yeah, come and do some house, what do you call it? Housekeeping. Yeah.
Yes, thank you so much, Prof. So I thank you so much for the final talk. And as we finish, I'll ask Onyancha to come and give a vote of thanks for everything that has occurred today, because we need to thank everybody who has been in this. Now, the whole aim of this symposium was for us to have a conversation, 1, amongst ourselves, 2, amongst other collaborators. So this is the beginning. I'm hoping that we can impress upon ourselves to organize another one next year, so that now we see how much we are moving step by step. It is good. Right now, Donana Mwago akona a very nice program there. Yeah, na nimeonyesha chakula. You get that? I had not known that. Niliona aliwin na award hapo hivyo na nashinda. What was it about? I'm just realizing that now. Equally, some of you, the nanotechnology, nanobiotechnology that you're working on. Mika, your computational chemistry. I knew Mika to be an analytical chemist. Now he's telling me about computational chemistry. There is no avenue that we have where we are communicating. So this is the first avenue that I hope we can now be able to stand upon and move forward. The process of organizing this thing, it's always tedious. You always have to go and beg people. You have to